Hey, it's kind of important. Let's talk about why. The first thing we want to talk about for DNA is its structure, because remember, structure determines function. So we want to first understand what the structure is so we can understand where that function comes from. DNA was discovered by a team of Francis Crick, James Watson, Maurice Wilkins, and Rosalind Franklin. Of course, there's more people involved whenever you're doing a complicated endeavor, though it was published in 1953 with just Crick and Watson. And they're the people with the Nobel Prize. There's a little bit of debate here about exactly why Rosalind Franklin was not included in the Nobel Prize. Part of it is because she unfortunately died of breast cancer and the Nobel Prize isn't awarded posthumously, but certainly she did not get nearly the same level of recognition that Crick and Watson did. And well, why? Sexism. Anyway, that aside, let's talk about the science part of it. Rosalind Franklin is the wonderful person that took this picture. This is X-ray chromatography. So if you expose things to X-rays, you can get some pretty pictures out of them. And this was how a lot of scientists were trying to figure out the molecular structure of different things because it's hard to look at something that's quite that small. Of course, this doesn't mean anything to people who are not well versed in how to interpret these pictures like myself, but this was the clue that let us know that DNA was a double helix. So let's talk about what that means. DNA, of course, stands for deoxyribonucleic acid. You can see here it is kind of in a ladder. So we have two strands with these base pairs in between, and then that ladder is twisted. You might also notice that the twist is not even. We have a major groove, that's the big space, and then a minor groove, the smaller space. So make sure whenever you are drawing DNA, it has this kind of irregular twist. It's not an even twist. And unfortunately, you see that depiction of DNA most of the time where it's not drawn correctly. That makes me grumpy. Another thing to notice is the strands themselves run in opposite directions. So it's a little bit easier to see in the untwisted ladder here, but you'll see 5' at one end and 3' at the other, and then for the other strand, it's just flipped. So it's kind of cool, they just run in different directions. And that has some interesting consequences for different DNA repair mechanisms and for how DNA is copied. Another thing to note is that it twists to the right. So a good way to think of this is take your right hand, this is my right, twist it to the right, but then also move it forward, and that is the way DNA twists. Let's test yourself. So which one is a right twist? That would be the correct form of DNA. If you guess the first one, that would be correct. Of course, if you're not drawing a super detailed picture of DNA, this doesn't really matter, but if you do want to make sure you have an accurate depiction of DNA, make sure it always twists to the right. So let's go back here. Other things to note, it has a sugar phosphate backbone. So in our diagram here, that red side of the ladder, that is where the sugar phosphate goes. It's sugar phosphate, sugar phosphate, sugar phosphate, all the way down. And then the nitrogenous bases in the middle, they are connected through hydrogen bonds. If you remember back to chemistry, hydrogen bonds are actually fairly weak. But if you have a whole bunch of them together and then you twist it on top of that, it does actually create a fairly stable organism. So what I like to imagine is we have two fairly solid strands, and in the middle, they're just connected by a bunch of magnets. And, not super strong ones, just normal magnets, not those fancy neodymium ones. But if you have a whole bunch of magnets in the middle and you twist it, all of that together creates a pretty stable molecule. Here is a nice way to summarize the chemical structure of DNA. So you can see the sugar phosphate backbone in detail. Then we have our 4 different bases, adenine, thymine, guanine, and cytosine. And then we have those hydrogen bonds that hold opposite base pairs together. Let's think more here though. Remember that structure is the key to function. It's the fundamental principles of physiology and molecular biology. And it is because DNA is stable that it makes a good storage medium. So this is our first great example, why structure determines function. And it allows us to store genetic material throughout an individual's lifetime, but also pass it on to our offspring. And it's because we can reliably make these copies that we are able to do this. But let's talk about those nucleotides. We have two versions here. We have purines and pyrimidines. Purines have two rings. These are adenine and guanine. Pyrimidines only have one ring in their molecular structure. Those are thymine and cytosine. So you can see them here. Notice adenine and guanine, they have two rings. They have one, six hexagon, and then they have one pentagon. And then thymine, cytosine, and uracil, that is only in RNA, not DNA, all of those only have one ring. So again, purines at the top, pyrimidines at the bottom. One really cool consequence of this molecular structure is called complementary base pairing. A purine will always bond with a pyrimidine. You can see in this diagram here, we always have purine with a pyrimidine, so we basically have like three rings as the width of a strand of DNA. You can imagine it wouldn't work if we had two purines together or two pyrimidines together, because they would either be too wide or not wide enough to bridge that gap. We also see consistent base pairing. Adenine always pairs with thymine, and guanine always pairs with cytosine. This is because with their shapes, they match together to form those hydrogen bonds in a specific way. And this is what allows for that really consistent DNA replication, because adenine will always pair with thymine, and guanine will always pair with cytosine. So if we have only one strand of DNA, we can make a copy quite easily. Here we can zoom in on the molecular structure here, so you can see this is why they have to bond this way. It just does not work if you take any two other base pairs and try to make them work. Here you will also see guanine with uracil and adenine with uracil, because uracil is in RNA. RNA is a little bit different. So let's talk about that now. RNA is ribonucleic acid. It has a slightly different molecular structure. It has one more oxygen. And this is primarily single-stranded. And again, we have uracil instead of thymine. It's just slightly different molecular structure. What's interesting about RNA, though, is we have so many different types. And because it's single-stranded, it can actually self-organize into complex three-dimensional structures. And unlike DNA, it does stuff. So here we can compare them. DNA is our deoxyribonucleic acid. RNA is ribonucleic acid. So we have, one more sugar in our ribose here, and we have uracil rather than thymine. But cytosine, guanine, and adenine are shared between the both of them. Here are a couple examples of the many different types of RNA we can find. If you've taken high school biology, you've probably heard of tRNA, rRNA, and mRNA before. So our transfer RNA, messenger RNA, and ribosomal RNA. But we also have a lot of microRNA. We have things that form other complex shapes, and just so many different types of RNA that I don't even know what it means, because this is not what I study. I feel like every time I check in with the RNA world, there's something new and exciting that someone has discovered. So RNA is a really complex and interesting thing, because RNA, we don't actually need to transform something into proteins to get an interesting and useful thing in the world of molecular biology. Because RNA is so varied, it has actually led to the theory called RNA world, that we think that before DNA was used as the hereditary material, its predecessor was RNA, because it can carry out so many of these functions. It's not always necessarily that efficient, but the fact that it can cleave bonds and create bonds depending on what type of RNA you have, that allows it to create the first instances of many biological functions. So what we think is that we had RNA that was able to self-replicate and eventually catalyze different proteins. And then it was eventually able to form slightly more complex structures. And once we got to the first cells, that's when DNA started to become the master template. When you compare the two, DNA is actually kind of boring. It doesn't really do much. It basically sits there and is stable. So that's why it's a really good storage medium, and that's why RNA does so many more interesting things and acts as the medium between DNA and all of our proteins. Chromosomes are, of course, super compacted DNA. So here is a nice little infographic. We start with our double helix, again with that uneven curve or grooves with our major and minor groove. We wrap them around our nucleosomes. Then we compact those nucleosomes together. We loop it into chromosome loops, and then we compact that even more into our chromosome. So chromosomes are super, super, super compacted DNA because our genomes are actually quite long, so it does take a lot of work to fold them up so they will fit within the nucleus of a cell. And again, here is a karyotype, so you can see all of our DNA is compacted into chromosomes here. Our DNA is actually loose in the nucleus for most of the life of the cells. Then we call it chromatin. And it is only compacted right before cellular division. So whenever you see one of these pictures like this, someone has treated these cells with a particular agent to induce cells to divide, and then has taken a picture and is hoping to capture a cell at the right moment where all of the chromosomes have already formed. So, what is the structure of DNA?